Good morning, good afternoon, and good evening. We welcome you to the first QPS webinar. My name is Robert Wronski. I am responsible for BD and marketing at QPS Austria. Conny Schweinzer, one of our scientists, is a molecular biologist and experienced with in vitro and in vivo studies. She will give now a presentation on an in vitro screening and the subsequent proof of concept study in one of our transgenic models. After the presentation, there will be a Q&A session where you will have the possibility to address questions to Conny. To do so, already during the presentation, please type your questions in the chat window. In case you do not see the window, please click on the small white arrow in the red rectangle in your webinar navigation. Please use the smaller window there to type your question. We will get back to you then after the presentation. You will have the opportunity then to ask your questions directly.
Thank you, Robert, for this nice introduction, and a warm welcome to everyone. My name is Conny, and I'm pleased to guide you through our first webinar entitled Early CNS Drug Development from In Vitro Screenings to In Vivo Proof of Concept. First of all, I would like to introduce the QPS Neuropharmacology unit to you. It is part of QPS Austria, and we are working in close collaboration with our colleagues of Netherlands. QPS Austria consists of two branches, a late-stage clinical research group and the Neuropharmacology unit. This first webinar, however, reflects what our Neuropharmacology unit represents: profound knowledge of and expertise in the field of neurodegenerative diseases. This figure illustrates the process of early drug development. It was chosen on purpose to stress out the complexity of this process. To cut a long story short, in 80 drug development projects, it is the major aim to discover promising lead compounds. As presented here, 80 drug development can be divided into several stages. The first one is phenotype modeling. It is all about designing assays that combine the interaction of the drug with the core element of the disease. For that purpose, you need a compound library. Since out of thousands, typically only one turns out to be a lead compound. Within the screening process, useful information is obtained and allows to draw conclusions about the drug target. Finally, the in vivo model itself represents the translational step that brings us closer to our aim to find a lead compound. If once a lead compound is found, clinical trials would be the next step. As mentioned before, QPS is also able to conduct clinical stage phase studies 1 to 4. Thus, we would be able to accompany you from the very beginning to the very end of your drug development project. This slide represents a simplified version of the previous one. In general, you need 3, let me call it ingredients, to perform the in vitro screening to in vivo proof of concept study. The first one you need is an agent that causes something. In our case, we highly desire a compound that would be able to alleviate neurodegenerative disease symptoms. These compounds can be either of natural or synthetic origin. Natural ones could be plant extracts, whereas synthetic ones like DAPT, a gamma secretase inhibitor. Secondly, you need a system where the agent can cause something. This can be a test system, in our case a cell-based assay. Since we have a very strong customer focus, we can offer you either primary cells or cell lines, depending on your requirements. And after an intense in vitro screening process, the next goal would be the in vivo model to really prove the concept. At QPS Austria, we have a huge field of assays or models we can offer you. I just listed some of them, for example, Alzheimer's disease, Parkinson's disease, Huntington disease, ALS, and Niemann-Pick type C disease. So we, together with our customers, then choose the adequate animal model to verify the hypothesis formed based on the previous in vitro screenings. Next, I would like to focus on the in vitro screening scenarios. This scheme shows the simplified version of the whole scenarios. So we start with a relatively large number of compounds within the first screening process, going down until very few of them, which then should go into the in vivo proof of concept studies. So preliminary, a really vast number of compounds are screened to evaluate beneficial or toxic effects on customized cellular systems. I will come to that later on in detail. Promising candidates of the first screen are then re-evaluated within second dose-dependent studies. And finally, indication-specific assays could reveal further hints towards the target of the compound. As promised, within the next slides, I will show some examples to further demonstrate the relevant steps. This slide demonstrates the first screening scenario. I again simplified it, since I mentioned that normally thousands of compounds are screened. Here I only displayed 29. In this previous scenario, we used primary hippocampal red neurons that were grown in the presence of pre-aggregated A-beta and the test compounds at the same time for a period of 6 days. After that, cell viability assays were performed, and what we can see here on this graph are the results obtained. The black bar on the right-hand side represents cells that were not lesions, so they did not receive any A-beta. These were just primary cells. The graph on the left-hand side next to it represent the lesion control, so these cells did only receive A-beta pre-aggregated, and the other gray bars from numbered with number 1 to 29 represents the cells that received A-beta together with the compound. And the field between the two dotted lines represents the so-called therapeutic window, which allows a discrimination between compounds that seems to be promising and able to increase cell viability, as for example this compound, number 18, in contrast to that compound which do not have any beneficial effects, as for example 21. So the next experiments should confirm these first screening results, and are shown on the next slides. So the number has already been reduced to 10 compounds only, and the setup was identical to the first one, since it should confirm the first results. But however, here we used two different doses of the compounds. We have a low dose and a high dose. And within this assay, we would like to see whether higher doses of the compound could even improve the rescue of the toxic effects of A-beta, as for example for compound 3 and 5 this scenario was true, and for others, like for example compound 10, the higher dose seems to be toxic to the cells. So as mentioned before, the third screening experiments should then be more indication-based and are displayed here. So if we again look on the compound number, we are already reduced to 1/10 of the starting number, so we only have 3 compounds left. And what we did then, we incubated the A-beta itself with the compound, here called X, Y, and Z. And what we did then was we performed an aggregation profile to see whether these compounds could inhibit the A-beta aggregation process itself. And as you can see here, Y and Z indeed were able to reduce the A-beta aggregate formation compared to the black bar, which represents the A-beta only, whereas X did not show these results but showed slight tendency to increase A-beta aggregation. The next experiments we performed was we incubated primary cells with the mixture mentioned before, so the A-beta together with the compounds. And what we saw was that, again, compound Y and Z, or Z, added to the cells could rescue the toxic effects of the A-beta itself, whereas X, again, did not show any significant effect. And on this microscopic picture, you can see on the very left-hand side the vehicle cells, which did not receive any A-beta. So they seem to be happy, they are viable, they form neurids, whereas the A-beta 42 causes toxicity to the cells. And finally, A-beta together with the test compound had rescuing effects again. So what we can see, in summary, we saw reduced neurotoxicity due inhibited A-beta aggregation. Thus, we considered that an APP in vivo model would be most suitable to test whether the compounds are able to counteract the amyloid-induced pathology and behavioral changes related to that. And therefore, we thought that an AD in vivo model would be the best and ideal test system to prove our concept in vivo. The next 2 slides will illustrate our APP Swedish London transgenic mice. They develop progressive increase in amyloid plaque burden over the age, starting at 6 months. Picture A demonstrates how a typical plaque looks like. You can see 6E10 positive staining combined with the thiol-S, which stains the core of the plaque. The mice also develop in parallel neuroinflammatory signs, so they have GFAP positive staining and CD11B positive staining. And since this presentation mainly focuses on A-beta, I would mention just aside that in figure D, you can see that these mice also develop or accumulate phosphorylated tau protein. Even more interesting, these mice also have behavioral deficits, starting again at 6 months of age, and the situation gets worse to the age of 9 months. So they have impaired spatial learning. Since we now have chosen and explained our in vivo model, we should consider next the experimental setup for the in vivo situation. As mentioned before, APP Swedish London mice develop AD phenotype at 6 months of age. Thus, we decided to choose 7-month-old male mice only, and they should be treated intraperitoneally weekly with compounds X and Z for the period of 2 months. After that, we would evaluate behavioral analysis as Morris Watermaze for the spatial learning. And after the end of the in vivo phase, histological and biochemical data should support or should reveal whether the compound has any beneficial effects or not. However, before performing efficacy studies, you always should consider about a PK study first to clarify important issues. Mainly, you should check whether your compound concentration in the plasma is high enough or whether the compound is able to enter the brain, since it should be a CNS drug. And if you're lucky, you can get such nice results as presented here, that the compound concentration in plasma negatively correlates with the A-beta levels in plasma. That should mean the more compounds you have, the less A-beta you can find in these animals. Here I already present the results we obtained. If we concentrate on the left-hand side, we can see a hippocampal section of mice treated with the vehicle. So we see large plaques in the vehically treated mice. Compound X, if you can remember, did not show any beneficial effects, and this is reflected also by this picture. The plaques did not disappear and did not get smaller, whereas compound Z again showed beneficial effects. The plaques were smaller and there was a smaller number of plaques in these hippocampal sections. The biochemical data showed similar results. Compared to the vehicle, compound X did not reduce A-beta 42 levels in the brain, whereas compound Z again significantly decreased A-beta 42 levels in these mice. And finally, the Morris Watermaze results showed an improve in the behavioral deficits in mice treated with compound Z compared to mice treated with compound X or the vehicle itself. So to conclude our results, we could prove that the compound Z reduced the A-beta levels in the brain, accompanied by decreased 6E10 immunoreactive areas, so reduced plaque size, and the behavioral deficits improved in this mice. So, as in vitro, compound Z inhibited A-beta aggregation in the APP Swedish London mice, so the proof of concept was achieved. Although this presentation focused on AD mainly, I would like to mention that we have several models that we can offer to you, which cover also other issues like Parkinson's disease, Huntington's disease, ALS, and many others. And in the end, I would like to thank you for your attention, and I would be pleased to answer your questions.
Thank you, Connie, for giving the presentation. We will start the Q&A session now with some questions that were asked by several participants. One question that was raised was how many animals per group are needed as a minimum number to perform a reasonable proof of concept study?
That's a good question. Of course, first of all, it always depends on the customer's needs and the readout we want to achieve. But during our long-term experience, we can say that a minimum number of 10 to 15 animals is sufficient.
And another open question is the following. Which treatment routes can be performed with your mouse models in the QPS facility in Austria?
That's also not easy to answer. Of course, it again depends on the customer's needs, but we are able to perform generally all application routes. So we can do oral gavage, we can do intraperitoneal application, we can do intrathecal application, intranasal, chronic infusion. It really depends again on the customer needs and on the bioavailability of the drug, of course.
Now there is a question about the in vitro part of the study. How did you measure cell viability in the cell culture model that was used?
In this specific experiments performed, we did MTT assays, but again, depending on the customer's needs, we can offer other assays as well. For example, what we did was urate outgrowth assays and synapse density assays.
You showed that in the APP model used, phosphorylated tau protein was seen also. Does QPS offer also specific models for tau pathology?
The answer is yes. You can also see it on the screen presented here. We have a mouse overexpressing the longest isoform of tau. We call it TMHT. And it carries the tau protein, or the tau gene carries two mutations, two missense mutations. And these mice display motor deficits. No motor deficits, but cognitive deficits.
In the study described, you used male animals only. Why was that done in that way?
We've seen that in some animal models we have sex-specific difference of the expression levels. This was also true for the APP Swedish London mice. So we have chosen male animals only to reduce this variety caused by the sex of the mice.
Thank you. Before ending the webinar, I would like to point out that we will be happy if you fill out the short survey that will appear after the session, and the other option to fill it out is to do it in the email that will be sent to you shortly after this session. Filling out the survey will take between 3 to 5 minutes only. Thank you for that. We herewith thank you for attending, close the webinar, and wish you a successful rest of the day. Goodbye.